Now in our cells, and we've described some of the cell cycle, we want to basically talk about, okay, what sequence of events does a cell go through in its, quote unquote, in its lifetime? That there's certain times, okay, we can do the same thing with us, where we say, okay, during this period of my life, I'm going to kindergarten. And in this period of my life, I'm in grades 1 through 6. And in this period, I'm in junior high. And in this period, I'm in high school. And in this period, okay, I'm playing like I've never played before. And in this period, okay, I have children and now I don't have any money because that's where all that goes to. So we have these periods in our lives that certain things are happening. Well, the cell cycle has the same thing where there's stages within the cell, okay, we call it interphase, where it's doing its cellular thing. But once it decides, hey, it's time to divide, so it's prepared to divide, it's gone through G1, the S, and the G2 phases that within interphase that is prepared to divide. And as it goes through that division, okay, now we do have to describe what type of cell is this. In our body, if we generalize our cells, we have what we call our body cells or our somatic cells. Somatic cells divide by the process of mitosis. We then have our germ cells, also called our gametes or our reproductive cells, so we're talking sperm and eggs. Those cells divide by the process of meiosis. So mitosis, somatic cells, body cells. Meiosis, okay, reproductive cells. So as we talk about the stages of mitosis, we're saying that we're talking about the stages of cell division. Now I'm going to be more specific here. The stages of nuclear division associated with somatic cells or non-reproductive cells. And the stages within those are prophase, metaphase, anaphase, telophase. So what you want to be able to do and what your objective asks of you is it asks you to describe each of these stages within mitosis. Again, mitosis being the nuclear division and the nuclear division of somatic cells. So what you want to be able to do is you want to be able to take each of these stages and whether we give you a graphic that depicts the stage, you need to be able to identify it. Whether we give you a description of that stage, you need to be able to identify it. So whether we do it in words, whether we do it in graphics, please be prepared to do that. So with each of these, we can list, all right, what's going on? Okay, what's going on in these stages? Okay. So again, mitosis is the actual active division of the cell. And we talk about interphase being part of the cell cycle, but it's not part of mitosis. Because again, looking at our big cell cycle graphic, all of the violet or purple color is interphase. It's down here in this piece of the pie, okay, the cell spends a small amount of time in its life actually dividing. So these would be the prophase, the metaphase, the anaphase, and the telophase. But interphase, where the cell's basically preparing for division and doing its cellular thing, okay, the G1, the G0, the S, and the G2 stages are within interphase. And so we're kind of showing them all side by side. So we want to be more specific, I mentioned, and that's why I reinforced it, that mitosis, by definition, is the process of dividing the nucleus. To finally actually get those cells to come apart as two daughter cells, two equal, containing the same amount of information, somatic cells, the term is cytokinesis, and I'll show you that process. So the first one, again, interphase, not a stage of mitosis, but it is a stage of the cell cycle, and it's everything except for the actual division. So if we really describe what was going on here, we would say, okay, the cell's doing its cellular thing, but we can't see it. One of the reasons we really can't see it is because we've taken that nuclear material, those long, big molecules of DNA, and we're using them. And so they're all spread out. It's kind of like, okay, this may be the only time scrapbooking is a great example of anything. Okay, just kidding. But you think when you get busy in a craft project, you fill the entire table with what you're doing. You got paper this and glue that and scissors this and Cricut that and stamp this and punch that, or whatever all those little devices are that people use to make those very creative pages. Okay, but you spread them out everywhere. Well, the cell does the same thing. The nuclear material, still within the nucleus, is spread out. And so we have uncoiled, uncondensed DNA, so we call it chromatin. And because we're trying to protect that nuclear material, we have an intact cell membrane. So we have an intact cell membrane, we have the presence of organelles within the cytoplasm. The nuclear material is chromatin, okay? Not condensed, not coiled up as chromosomes, and we're going to get into those here in just a minute. All right, so as it begins the process of mitosis, well, now what do we see? Well, now if we look at the nucleus, it's starting to, or the nuclear membrane specifically, it's starting to break apart. And in the process of interphase, we took our centrosomes and we duplicated them so that we're going to get one of them going to one pole and one of them going to the other. Because what we need to do is take all of this information in the middle of the DNA and we're going to pull it apart. And we're going to send half of it one way and half of it the other. Okay, so as we look at our nuclear material, notice up here in interphase, it was just kind of this granular appearance. There really wasn't a whole lot to see. But down here, we have strands of stuff, okay, that's actually present there. So up here, it was chromatin. Down here, they're chromosomes. Now, one of the things that I think is very important to mention at this point is whenever we draw chromosomes, so if you asked anyone who has any biology experience whatsoever, say, okay, draw me a chromosome. Okay, probably of those who have studied it before, 99% of them are going to draw it like this. And they're going to draw it to look like an X. And we do that, and notice on the graphic, okay, they look that way as well. Because in that S phase, in that process of replicating DNA, we took one big long molecule of DNA and we replicated it. And we replicated it and we held those two strands, the one that was the original and now the new one, okay, and we held them together. We held them together, okay, at a centromere, so the connection of the two strands. Now this is the one time we use the term chromatid, because when we make these chromosomes, and we go to my graphic, okay, when we form these chromosomes in preparation for division, we have one chromatid and one chromatid. Okay, so we have two total chromatids, and because they have identical information, because this strand and this strand coded from the same information, but we want them to be the same, because we're going to split this thing apart. And half of this chromosome is going to go to one new cell and half is going to go to the other. Well, they got to get the identical information. We can't send information into one cell that's different than the other, or they would function differently. Well, if they're supposed to be identical cells, we can't have them function differently. So we have to distinguish the two strands from each other. So this would be one chromatid, and this would be one chromatid. So while they're connected here in the middle is the one time we refer to the entire hunk of genetics. And maybe think about it that way. Whenever I have a hunk of genetics by itself, that is a chromosome. And that'll help make sense of some other stuff later. So when I have a hunk of genetics, that is a chromosome. When I have duplicated strands of DNA in a chromosome that are connected at a centromere, I have two sister chromatids. And the reason I bring this up is because when I split these apart, I end up with a chromatid going this way, a chromatid going this way. Okay? But now they're hunks of DNA again. This is one hunk of DNA, and this is one hunk of DNA. And so this is where science confuses us a little bit, where it says, okay, I had a chromosome that had two sister chromatids. I pulled that chromosome apart and got 2 chromosomes. You're like, wait a minute, what do you mean? When I had chromosomes here and I have chromosomes here? I think the easiest way to think about this is whenever I have a hunk of DNA, I have a chromosome, whether that hunk is big or whether that hunk is little. And that's going to help us keep track of these numbers. Okay, so let's look at, going back to our slide, let's look at the hallmarks. So we said with interphase. With interphase, I have chromatin, not chromosomes. I have an intact nuclear membrane, okay, and I'm going through my cellular thing, the main life of my cell. In preparation for division, we move on to prophase. In prophase, the centrosomes containing the centrioles are going to be migrating to the poles of the cells. That means one side and the other. the nuclear membrane begins to fall apart. It's kind of tough to pull nuclear stuff apart if the nuclear membrane's in the way. And so extensions, microtubules from the centrioles, are going to form what we call the mitotic spindle. And those filaments are going to attach at the centromeres of the chromosomes, and that's what's going to help pull these apart. So I've lost my nuclear membrane, I've got chromosomes, not chromatin, and I have duplicated strands of DNA, and that's why it has the appearance of the X. Now the next one's the easiest. The next one's the easiest, metaphase, because M stands for middle. And so this is when our chromosomes all try and line up in the middle so that the mitotic spindle can attach to them. Now this has a couple of advantages. One, they're all together, and we can pull them apart effectively. And number two, we can take a picture of them. So just like, all right, let's say you're going to, because everybody has to, gets the opportunity to go to a family reunion. And at those family reunions, no matter when you come, no matter when you go, there's some point that somebody's going to bring out the camera and they want everybody to get together so that they can take a great big family picture. They want to take a 4-generation picture. They want to take a 3-generation picture. If you have genetic defects in your family, nobody sticks around very long. It could be a 2-generation picture. That's not a funny joke, but a little bit. But they want to take a picture of everybody. Well, they can't take a picture of everybody, if everybody's spread out all over the park. So they make everybody pouts about it, but everybody comes together and squishes as tight as possible. So the advantage during metaphase is the chromosomes are about as close together as they're going to get. And later in metaphase, they're getting pretty tight in terms of being condensed as strands of DNA. And so it's a great time to take a picture. Now, they're not all lined up perfectly, and so it does take a little bit of dividing up these chromosomes to line them up. And so what happens is we take a picture of the chromosomes during metaphase, and that ordered display of chromosomes that we create is called a karyotype. So a karyotype is an ordered display of chromosomes where we can look at them. Now, what we want is in our somatic cells, we want there to be 46 of these. We don't want 45, we don't want 47, we don't want 48, we want 46. So it's just like when you buy something and you have to assemble it. The worst part of assembly is when things don't go right. And if you think about assembling something, you have 3 outcomes. Outcome number 1, went together perfectly, because we read directions, but as men, we don't read directions. The other 2 options. Option number 1, there were missing parts. Option number 2, there were extra parts. And so now you start thinking, well, where did I miss something? Because there's not supposed to be any extra parts. Well, the same thing's true here. If we are thinking that there's a chromosome problem in terms of chromosome number, well, we're supposed to have 46 of these, but darn it, we don't. We have 45, or we have 47, or we have 48. And so we want there to be 46 of them. And so what we do is we line them up. And notice we line them up from biggest to smallest, and then the sex chromosomes, the X, and in this case an X and a Y. So we know the gender of this person. We know the gender is male because they have an X chromosome and a Y chromosome. If we had 2 X's there, of course, the person would be female. Now, are there situations where there can be 2 X's and a Y? Yeah, it's a condition called Klinefelter syndrome. Is there a condition where they only have an X there? Yeah, it's called Turner syndrome. Is there a condition that there's only a Y? There isn't. It just so happens you can't live without the X. Ladies, don't let it go to your head. But it's true. So in that scenario, we line them up, biggest to smallest. We also look at them related to the position of the centromere. Where is it? So that they're similar in size. We line them up by where they're attached. The top part of the chromosome, usually the shorter part, is called the short arm. Bottom part's called the long arm. So we have the short arm of the chromosome, we have the long arm of the chromosome, we have the centromere where they attach. So we have 2 of them. Well, you have 2 chromosome 1s, 2 2s, 2 3s, and you got them from your parents. So one of your chromosome 1s you got from mom, the other one you got from dad. And it just so happens that these chromosomes have essentially the same genes on them. You're like, there's no way. There's no way I have the same genes on this chromosome 1 I got from my dad and the one I got from my mom. There's no way for that. Well, you do. They are the same genes, but they're not the same instructions on that gene. So what I mean by that is, if I said that a segment of DNA on your chromosome 9 coded for your blood type, that means everybody has, we can never say always, but everybody has a blood type gene on chromosome 9, determining whether they're of the A blood type, the B blood type, or the O blood type. Everyone's got a blood type gene, but they may have a different version of that gene, and we call those alleles. So you may have the A allele of the blood type gene, the B allele of the blood type gene, the O allele of the blood type gene, but you got a blood type gene. So the genes on chromosome 1 are the same as the genes on the other chromosome 1, and the same thing is true of 2s and 3s and 4s and so on. But those instructions, the alleles that are there may be slightly different, and that'll come into play with some of the stuff we talk about later, but understanding that. So what we do with these karyotypes is we look at, are there the appropriate number of chromosomes? When you hear terms like trisomy, having 3 of a given number of chromosomes, monosomy, having 1. Okay, one of the common ones most students have heard of is trisomy 21. Trisomy 21 being that there are 3 copies of 21, and that's our leading cause of Down syndrome. And we'll get into that process when we get into the reproductive unit. You kind of get the idea there. All right, so our next one, and we'll come back to genetics a little bit later. Our next one, anaphase. Now, notice what's happening. I'm starting to elongate the cell. I now have pulled apart the chromosomes into chromatids, and they're being pulled to opposite poles by the mitotic spindle. That's what's happening in anaphase. As we move to telophase, notice that we've pulled the chromosomes to opposite sides. So we've split those duplicated chromosomes into individual strands of DNA. We've reformed the nuclear membrane, and now we're pinching off. And so we've got this cleavage furrow. We've pinched off the cytoplasm, and so we finish the process by dividing the cytoplasm through the process of cytokinesis. And that's why I said earlier that division of the nucleus is mitosis. Finishing that division and forming 2 identical cells by division of the cytoplasm, okay, is cytokinesis. So that's the difference in terms of those relationships. Then once we're done with cytokinesis, we revert back to each of these 2 new identical daughter cells, okay, revert back to interphase and do their cellular thing.